Hi, I'm Josh Reeves, and here I'm presenting our work within the Habib Lab at King's College London in developing transplantable Wnt-directed osteogenic human organoid bandages to improve bone repair. Wnts are a family of signaling proteins integral to many aspects of development, growth, and homeostasis. Within this, they have a particular role in tissue patterning. Due to post-translational palmitoylation, Wnts are hydrophobic, and in many cases are secreted locally, and as a result are presented directionally to receiving cells. Wnts therefore can confer spatial information, and are known to mediate asymmetric cell division both in vivo, such as in C. elegans, and more recently in vitro in mouse embryonic stem cells. To enable the study of localized Wnt in adult human stem cells, we generate a localized Wnt source by covalently immobilizing purified Wnt3A protein onto polymeric surfaces, thereby recapitulating the directional Wnt presentation of the in vivo environment. Human skeletal stem cells can be isolated from adult bone marrow and are able to differentiate into bone, cartilage, and adipose tissue. 53% of these cells are Wnt responsive, and when supplied soluble Wnt3A, they proliferate in a 2D in vitro culture. Human skeletal stem cells that express high levels of stem cell markers and low osteogenic markers are seeded onto this Wnt3A surface and overlaid with an osteogenic collagen gel. At mitosis, they orient their spindle towards the Wnt3A surface and become polarized. Skeletal stem cell markers such as STRO1 and CDH13, shown in cyan, segregate towards the half of the cell in contact with the Wnt, whilst the Wnt distal half has a higher ratio of osteogenic sulfate markers such as osteopontin and osteocalcin, shown in magenta. The resulting asymmetric cell division produces one self-renewed stem cell that retains Wnt contact and one cell undergoing osteogenic differentiation that enters and migrates through the 3D gel. The polarization at the single cell level influences the polarization of the entire organoid. Within 7 days of culture, a spatially defined cascade of osteogenic differentiation is formed, with stem cells at the Wnt surface and osteogenically differentiating cells throughout the 3D matrix. This engineered tissue is named the Wnt-induced osteogenic tissue model, or wyotome, and crucially, without the localized Wnt source, the structure does not form. As it recapitulates the entire osteogenic environment, the wyotome has great potential for the in vitro study of human osteogenesis. We next asked whether the effects of localized Wnt and tissue engineering can be applied to in vivo bone repair. We immobilized Wnt onto flexible, biocompatible polymers, forming the Wnt3A bandage, and transplanted them onto critical-sized calvarial defects in mice, meaning that without intervention these defects would never fully heal. The defect without treatment, or those treated with inert bandage controls, showed only minimal healing. However, the Wnt bandage doubled the area of new bone. When the wyotome organoid is formed upon the Wnt bandage, and the whole structure implanted into the defect, the area of new bone was tripled. Looking within the defect area, as designated between the two arrowheads, through immunofluorescent lineage tracing, we determined that implanted human cells contribute to both the new bone and the stem cell population within the defect. But not only is the amount of repair increased, but the areas of new bone are histologically similar to mature bone. Lamellar mineralization, aligned osteoclasts, and the presence of blood vessels indicates well-organized tissue. This is an improvement over many proposed bone repair approaches, which promote crude osteogenesis at the expense of competent tissue structure. Altogether, the wyotome formation is driven by the Wnt3A-mediated asymmetric cell division of human skeletal stem cells, resulting in an engineered tissue that recapitulates the human osteogenic environment. In vivo, localized Wnt3A technology improves bone repair, and the combined cell therapy approach of the wyotome bandage further increases the formation of new, but crucially histologically mature, bone. Together, this demonstrates a significant progression in the long-term survival and in vivo differentiation of transplanted human skeletal stem cells, which may also have application for the study and treatment of other Wnt-responsive tissues. Thank you very much for joining me in this presentation. We welcome you all to come and discuss our project further and follow us on our website, habiblab.org, and on Twitter at shukrihabib_lab. Thank you very much.